Hey guys, so we didn't have time in class to talk about cell communication today, so here is our video. So cell communication, this is how our cells are going to communicate with other cells, and they do this through direct contact with the cells, okay? This happens in multicellular organisms, so we can send signals and messages to other cells in our tissues and then in our organs. It also happens in unicellular organisms that exist in colonies. So here we have a colony, and each of these little green dots is actually a different organism, and they are able to communicate as a colony to coordinate their movements. Okay, so we have some examples. You don't have to memorize these examples necessarily, but just get an idea of an example of this is our immune cells, where we have cell-to-cell contact, where we will have a cell that is a B cell that gets activated and is triggered when it binds to an antigen. Okay, so we have our immune cells recognizing some sort of pathogenic bacteria. They recognize it when they actually make direct contact with it. So that's just an example of where two cells are communicating. One is a pathogenic cell and one is an immune cell. Okay, and we have four different types of cell communication that we're going to talk about. One is autocrine. This is when a cell is communicating with itself and it's talking and sending signals back to itself. We have juxtacrine. Juxtacrine is when it's really close to and next to our cell. This is going to be through gap junctions, little junctions in between two cells that are next to each other. We have paracrine, where it's signaling another cell that is nearby but not directly next to. And then we have endocrine, and this is where a cell is going to target through a long distance, usually through the bloodstream. Okay, this is like our endocrine system in the body. Okay, so an example of a couple of these juxtacrine, this is when we have chemical cells directly adjacent to, and it could be through a cell membrane or cell wall if it's a plant cell. Okay, so for our cell wall, that's like our plasmodesmata that we've talked about in the past, those little gaps in the cell wall. Those are going to allow for signals to go through from one cell to the next cell. Okay, so that is going to be our juxtacrine signaling. And in the cell membrane, in that case, we have something called gap junctions, which, similar idea, are just little gaps, little connections between one cell membrane of one cell to the cell membrane of another cell. An example of paracrine signaling, when we have cells that are close to but not directly touching, are our neurotransmitters in neurons. Okay, so you might have known this example in the past, but we have exocytotic vessels releasing. So that means we're doing exocytosis here, releasing little packets of vesicles that have signal molecules in them, and they will go to another cell that is very close by. They can travel short distances across small gaps between the cells. Okay, and we have a video here showing that. Give it a second to load. Here we go.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Okay, so we don't have to actually memorize all of those little steps in there.
Communication between
the big point is that we have a signal being sent and it's not directly touching, the two cells are not directly touching, it's just being sent, and there are receptors on one cell. Okay, so then we also have endocrine signaling. An example of that are hormones. Hormones are secreted by glands, and that means that they need to travel from those glands to specific targeted cells. And so they will do this through blood vessels. This is our endocrine system that's going to produce them, which are different glands here, are the organ systems involved in the endocrine system that make different hormones. Okay, so all of these, though, are dealing with signal transduction, whether it's autocrine or juxtacrine or endocrine, all of these different types of cell communication, we use this process called signal transduction. And there are 3 steps to signal transduction. It's also referred to as cell signaling is another way to think about signal transduction. And the 3 stages are first reception. So how is that signal received by the cell? Okay, and it's the detection of the molecule coming from somewhere else outside. Then we have transduction. This is where we convert that signal to a form that can bring about a cellular response. Usually we are amplifying the signal so that we increase, so that we have one molecule, but then we end up amplifying so that multiple molecules are going out and sending this signal, transduction. And then we finally have some sort of response, so some sort of cellular response that's happening because of this new signal that we just received. So this reception, transduction, response is what we want to be able to look for. And we will be looking at some examples. For these examples, you don't have to memorize the specific steps, but you need to be able to recognize what part of it is the reception, what part of it is the transduction, and what part of it is the response. Because on the AP exam, they won't expect you to memorize one, but they will give you an example, and you would have to be able to identify these parts of that particular example. Okay, we have a video that's going to pop up. So we're not going to watch this whole video, but this is from a music video from OK Go, and it's a giant Rube Goldberg machine. So we'll watch a little bit of it. This is kind of the idea of our transduction. Anyway, I will let you look this video up if you would like to watch it. The point is at the very end of the video, what happens. Okay, so the cells communicate in similar ways. We have our reception. In this case, it was that little truck that was making the first movement. Then we have our transduction, where that signal keeps moving, and it's going to actually get bigger and bigger and bigger of a signal. And then we have some sort of response, and in this case, it was the paint being sprayed. Okay, so we're going to talk about these different steps. The first one is our signal reception. Okay, so there's different ways we can have the signal received by the cell. And a big word that you're going to need to know is ligand or ligand. That is going to be a chemical messenger, and it is what is going to be received by the cell. And it is specific to the receptor. Okay, so we have a specific ligand that will bind to a specific receptor in order to have some sort of response from the cell. And they're specific so that we don't have cell responses happening for just any reason. We have to have a specific molecule that makes a specific response happen. Okay, so some examples of this is we can have some molecules that are able to just go straight through the membrane when they get to the cell. So think about what that would mean. The signal molecule, that ligand, would be, and they would be hydrophobic, right? Because they're similar to the membrane. Hydrophobic molecules can go right through, usually small hydrophobic or even fairly large hydrophobic molecules can go straight through. So some signal molecules are hydrophobic, and that means that the receptor is going to be inside the cytoplasm of the cell. So that signal molecule, that ligand, can come through, and it can bond to the receptor. An example of this is steroid hormones. So that's because these are made from cholesterol, which you might remember is a lipid because of its it being nonpolar and therefore hydrophobic. And so from cholesterol, we can actually make things like estradiol, which is a form of estrogen, and testosterone. So these are examples of steroid hormones that are nonpolar, therefore hydrophobic, and they can go straight through the membrane and pass into our cells and attach to a receptor inside the cell. We can also have some cells that cannot go straight through the membrane, so therefore they're not hydrophobic, they are hydrophilic. So because of that, they're going to need either some sort of channel to pass through, or a lot of times we will have a receptor on the outside of the cell. Okay, so example of this are our gated channels, where we will have a channel that opens that molecule or some sort of receptor on the outside of our cell. Okay, here's an example of one.
There are a number of ways that ion channels can open up in the cell membrane. Here are two ways that are really important when understanding how a neuron sends signals to other neurons. The first one here is a chemical-gated ion channel, and it has a receptor site for a chemical. Now that chemical can come in and, let's say, a neurotransmitter and fit in the receptor. This will cause the receptor to open up. It's kind of like a key into a lock. Sodium can come rushing in and may affect another type of ion channel, but this is voltage gated. When the voltage around this ion channel gets to a threshold, a very specific level, then it too can open up and ions can come rushing in. So in this case, a chemical opened it up, and in this case, these positively charged ions came into the cell, changing the polarity around this ion channel and opened it up in that way.
Okay, so that showed
your site for a chemical.
How we can have a receptor come in and or we can have a ligand come in and attach to a receptor on the outside of the cell and create some sort of response. Okay, so our reception can also include, this is an example where we have something called a G protein, a G coupled receptor. So a G protein is involved in this receptor. So the big thing on these is this type of membrane protein, so similar to like those channels, and it interacts with an ion channel usually in the membrane. And these are called G proteins because they have something called GDP and GTP that they interact with, which is a very similar idea to our ATP, just instead of A for adenine, these are G, so they're forms of guanine, and they are involved in the mechanism, but the idea is that this protein can be used to signal some sort of response when it's activated. Okay, and we'll go into more details about how a G protein works next class. Okay, we can also have things like enzyme receptors. So this can happen where we have phosphorylation, which we've talked about, that adding of a phosphate, but that can also happen to a receptor outside of the cell. So we can have a molecule that signals some sort of phosphorylation occurring. Okay, and whenever we have a phosphate being added, that is going to make a conformational change, so a structural change to that enzyme, to that, in this case, it's bedded in the membrane, but it's going to make a structural change, and that would be some sort of reception example. So one thing that will do this is we have kinases, and kinase is a word that is just referring to an enzyme that phosphorylates something. So that's a word that we will need to know. So kinases are sometimes involved in this signal reception. Okay, so we want to think about what types of things get phosphorylated. So a lot of times the amino groups on proteins are going to get phosphorylated, and that's what's going to make that conformational change whenever we have a signal being received. Okay, and so think about what is the shape of our amino acid, which we've talked about in the past. And we have our central carbon, we have a side chain, we have a carboxyl group or carboxylic acid, and we have an amino group, okay? And so the part of our amino acid that's going to be phosphorylated is actually the side chain. So here is an example of the side chain here, the R group of a serine amino acid, and then we can just add a phospho group so to make phosphoserine. And this is how we would phosphorylate an amino acid on an enzyme. So that's the part that's actually being phosphorylated when we phosphorylate an enzyme. So there are several enzymes that are phosphorylated quite often. Serine is one of them, as well as tyrosine and threonine. Do you need to memorize these? No, but just know that multiple different types of amino acids can be phosphorylated, and that can, again, make some sort of conformational change in the shape of the enzyme and therefore change the function of the enzyme. Okay, so we've talked about how a signal is received. Now we're going to talk about signal transduction. Okay, so some examples of signal transduction pathways, a lot of them are going to involve what's called a phosphorylation cascade. Okay, and this is where we relay a signal through a bunch of phosphorylation happening. Okay, and this is enhancing and specifically amplifying our signal. Okay, so here we have something that's phosphorylating one thing, and then that phosphorylates something else, and then that phosphorylates something else, and that phosphorylates something else. And the reason this is a cascade is because it's getting bigger and bigger and bigger as we go out. We have one enzyme that phosphorylates, but that one enzyme can go on to phosphorylate many different other enzymes, and then each of those phosphorylated enzymes can go on to phosphorylate many other enzymes. So as we phosphorylate one thing, it's exponentially increasing the number of things that can be phosphorylated, therefore amplifying the signal that was received by one molecule, and it can go out and send that signal to the whole entire cell. Okay, and we can also have molecules that interfere with this that are phosphatases. So remember, we have that ACE ending is going to be an enzyme that actually removes phosphates. So remember, kinase adds phosphates, phosphatases remove phosphates. And these can actually stop some phosphorylation cascades or even make them decrease. Okay, so another example of our transduction is we can have proteins that are modified, and this is going to affect the regulation of proteins that are being synthesized, and also how proteins, their activity in the cytoplasm. So an example here where we have DNA that's producing these different enzymes, we can then turn off certain genes. The same idea is happening with signal transduction in this way. We can have a signal being sent by turning off genes that therefore impact what is being synthesized in the cell. Okay, so here is an example where we have a signal molecule that goes through the cell membrane. And in this case, we have a receptor that's inside the cell. So this is probably a hydrophobic molecule, and it receives the receptor, and that receptor can sometimes even travel with it, and it will travel and take this to the DNA, where we then affect what genes are being or are not being transcribed and turning into proteins. So this is another way we can regulate our another way that the signal is sent once it gets to the cell. Okay, this video has an example of this.
Aldosterone is a lipid-soluble hormone that can easily diffuse through the plasma membrane. Inside the cell, aldosterone binds with an aldosterone receptor molecule in the cytoplasm. The aldosterone receptor complex moves into the nucleus and binds to DNA. The binding of the aldosterone receptor complex to DNA stimulates the synthesis of messenger RNA, which codes for specific proteins. The messenger RNA moves from the nucleus into the cytoplasm and binds to ribosomes, where it directs the synthesis of specific proteins. These proteins produce the response of the cell to aldosterone.
Okay, so some of these signals can actually induce the synthesis of different genes in there for proteins, or it can either inhibit it. So we can have either one depending on the function of a particular messenger. Okay, so now we're going to get into our responses. This is some examples of some cell responses we have from signals. So one example of a response that a cell can have is apoptosis. And we have talked about this a little bit, but this is programmed cell death. And so once the signal is received and then we go through transduction and we have the cell responding, we can have one of those responses being that the cell dies and it goes through and shuts down. So this is actually what is creating digits on your hands and toes. So in development, we have seen, this is an example, I think of a mouse hand that's being formed, but these cells in between the fingers actually get signaled through a pathway that they need to go through apoptosis. So the cells that were in between these fingers are actually signaled and told to go through apoptosis, and that's how we are able to have spaces between our fingers. Otherwise, if we did not have this apoptosis signal being sent, we would have webbed fingers and toes, which is kind of interesting. Okay, another example of a response is for cell growth and division. So kind of the opposite of our apoptosis, where the signal is signaling the cell to die. Here we are signaling the cell that it's time to grow and time to divide. Okay, and actually having an error in this is where we get examples of cancer, where there's too many signals being sent saying that it's time to grow, makes us have tumors being developed, but we still need some signaling to have normal growth in our cells. Another example of this is with our metabolism. So we can have signals that say, hey, it's time to increase cell metabolism and time to use energy. Okay, so an example of this is insulin is a signal molecule that is going to signal for glycogen, which is made of, is a carbohydrate that's made of many glucose molecules, to be broken down into glucose that we can then use for the cell. Okay, something that affects this is adrenaline, or also called epinephrine. So this is a similar pathway as well. Okay, and so that is actually all we're going to talk about. We're going to go over some questions next time in class. Bye, guys.